So for the next aspect of the activities this afternoon, I'd like to have Dr. Holland, Dr. Lukacs, Dr. Castro, and Dr. Zou join us up on the podium. This is going to be a panel discussion. There are some questions we'd like to ask to stimulate some of the discussions with you all in the audience.
I think you have to walk. Oh, yeah.
Your mics are all on. They're taped.
So everyone be careful because the mics are hot.
Play with them. It's taped on, so you can't.
So we'd like to keep this fairly interactive. And the first question I'd like to pose to Weiping Zou. New biologics are bringing precision to oncology care and other inflammatory disorders. Where do you see these types of therapies making impact next, and what targets might be relevant, and what are some of the challenges?
Right. Can you hear me well? Yes, and we often say it's hard to predict. Only God knows what's going on next. But nonetheless, based on what we have so far, at least in the field of cancer immunology and immunotherapy, so far, we have three things going on. Number one, as you all know, that is a checkpoint therapy. Basically, we have some antibodies can block the immunosuppressive mechanisms. So at this point, this reaches the bottleneck. So we have about maybe 30% of clinical response so far. So what people are looking for, always, hopefully, we can find some novel mechanisms, novel immune evasion mechanisms. Of course, they should be. We don't know much yet, but since many investigators are involved, based on the current technology, we may be able to find some critical and important new checkpoints. So that's one way to go. So another direction in this field is CAR T-cell therapy. You all hear about this. People always predict maybe CAR T-cell therapy, such as Castro, will receive Nobel Prize, but somehow it's not there yet. Since immunotherapy received Nobel Prize in 2018. So anyways, so CAR T-cell therapy so far works pretty well in some specific hematopoietic malignancy, particularly CD19 positive, as you all know. It has a lot of space to go. A lot of engineers, protein engineers, chemical engineers, computer engineers, they are all involved in this space. So I have high confidence in this case. We may be able to find a way to go to treat some fractions of patients because the technology is really moving very fast. Another way is vaccine. I still feel vaccine should be a good way to go. It should have its future. At this point, vaccine really works in the infectious disease area as the main topic today. We all know this. But in the cancer field, we have been very much frustrated by early results we have. Early results means about maybe 15, 20 years ago when people used DC as a vehicle to do different vaccines. We didn't see much in terms of clinical response. But right now, since we are able to predict and to define and discover neoantigens in tumors, maybe we have a way to go. So in my view, those three areas will provide a lot of new discoveries. But nonetheless, the basic understanding means the basic science. It is always the driving force. I stop here.
Thank you. So does anyone have any questions about this? So I'd like to start by asking a question. Although CAR T therapy has really been directed at T cells, there is research with using other cells, such as NK cells, to perform the same activities. Is that going to become more common, that there are other lymphocyte populations?
Yeah, so when I give talks, when I emphasize T cells, and people always ask, so how about the other immune cells, which are also important, as you mentioned, the NK cells, right? So in theory, let's say evolutionarily speaking, we expect innate immune response and adaptive response work together. That's the best way we can go. And so far, actually, the vaccine works. At least part of the vaccine works. Why it works? I guess it's a collaboration between innate immune system and adaptive immune system work together. We need to find a way how we put, make innate immune system and adaptive immune system work together, such as NK cells, some innate immune signalings, what Michelle and other folks talked about, for example, interferon pathways, type 1 interferon pathway, type 1, type 2, type 3, and if they can all work together, probably that's the best way to go.
Great, thanks. Any other questions? You can have to just shout out. I can't see you with the bright lights up here. All right. Okay, well, if you do hold that question, you can ask later. So we have a question for Dr. Castro. How do you see factors like climate change or other changes in our environment impacting inflammation and health? And how do we best prepare for these potential changes?
Thank you. So when I first read that question, because of course we were told what questions were going to be asked, I almost collapsed because I said, "Oh, this is not my area. What am I going to say?" So I went into Google and I found a really wonderful website by a professor in Australia whose name is Jennifer Stobbe. And the title of that website is "The 21st Century It's an Inflammatory Century, and We've Created It Ourselves." So I thought, that really makes sense. And I thought, the website is really beautiful, and it talks about all climate change, weather, the sun, that we listened to several of the talks today, and how that may impact inflammation. So it goes on to talk about, for example, even cold weather and how can it affect inflamed joints due to psoriasis, and pollution and how that can impact lung cancer, asthma, respiratory diseases. So essentially it got me thinking that really, absolutely right, it's a wonderful question, and I do think that air pollutants and smoke and cigarette smoke and additives that we put in our foods and sun exposure really is a massive inflammatory stimulus to our bodies, and how do our bodies deal with it, and what is the impact of that? And in relation to that, I don't know if you are aware that the RNA Initiative showed a screening of a movie called Awakenings, and in that movie it talks about a pandemic, a very different disease, a very different pandemic compared to the COVID one, because it was a neurodegenerative pandemic. No one knows the causes of that pandemic, and it happened between 1915 and 1926 throughout the world. And it seems that in the US there were 1 million people affected and 500,000 people died. No one knows the causes of that disease, and it's a disease called encephalitis lethargica, and people just froze. Their minds were alive, but their bodies would not respond to what their brain was telling them to do. And again, that's a disease for which there's no cause was ever discovered. People thought, at one point, scientists thought that there could be viruses, but no viruses were related to the onset of the disease. So people now think it could be some climate or environmental factor that triggered this pandemic. So there's a lot of work that needs to be done in this area. And in terms of what we can do, I think as individuals we can do a lot, and I would like to hear ideas from the people in the audience here. We can drive less, we can use more public transportation, we can consume, there's a big movement now from farm to table, consume fresh products, less processed foods, and there's many other things in terms of controlling how corporations do their businesses to pollute less. We've heard about the fires in California, fires in Europe, extreme temperatures. So I think it's a very, very topical question, and I thank you very much for asking it, because it got me thinking about it and how it really can impact health and well-being of human race in a way. So it's a very big question.
Thank you, Maria. Any comments, questions from the audience?
I don't know if there's anyone working in that aspect of inflammatory at the University of Michigan.
Well, I'll punt it to Steve. Is anyone at the NIH working
Who's
on?
that?
Steve Holland.
Is anybody working on
On climate change infecting pathogens.
Pollution, temperature.
Yes, though not specifically in the climate change context, but I think it's hard to separate those out. We have one investigator in particular, Ian Miles, who's very interested in the role of pollution in atopic dermatitis. He's got some extraordinary data about the role of isothiocyanates in pollution, which are a major component of industrial pollution. And what he shows quite clearly is that the upsurge in atopic dermatitis in children is highly correlated with being in industrial areas where this particular pollutant is higher. So I don't think it's a climate change globally, but it's a climate change locally, and how that affects, you know, everything from microbiome to, and his interest is in how the pollution affects the microbiome, quite persuasive. But it really is a sort of an environmental interaction.
Yeah, thanks.
And I guess I'd like to make a comment, if I can, over here.
Please.
Yeah, so I'm John Osterholzer. I'm a pulmonologist with U of M and at the VA, and my research program studies deployment-related respiratory disease in veterans, and we're looking at ways in which, like, exposures to military burn pits, massive sand, dust storms, you know, huge amounts of fuel and fumes impact respiratory health. And I think one of the challenges we're really seeing is that this is like studying a new disease, right? Our immune system evolved over, you know, tens of thousands, hundreds of thousands of years with sort of a set number of stimuli, a set number of pathogens, things like that. But, you know, now our lungs, our world is just bombarded with entirely new types of stimulants. Like, who would ever think that burning a bunch of garbage and having people like inhale this, you know, right in close proximity is a good idea? Where we think about the, you know, the 9/11 bombings and how a massive building exploded in the context of jet fuel that was the combustion source for that. Where you think about wildfires now, which aren't the wildfires of old that, you know, our immune systems might have been accustomed to, but they're wildfires that are, you know, filled with all kinds of homes burning up and all other sorts of industrial things being in the mix. And so I guess one of my comments is, yes, there's people studying it, and it's proving difficult because you're trying to essentially try to put potentially new diseases into boxes that people think they understand, and when they don't fit, people don't know, you know, how to explain them, how to understand them. I guess my question for the panel might be, you know, how do we study diseases that are from stimuli that our immune system really never saw before? These aren't necessarily pathogens, they're complex composition of all types of metals and gases and other sorts of things, particulate matter. Should we be thinking about things differently? Should we be thinking about, you know, not necessarily dysregulated immune response, but simply a very highly regulated system's response to something it hasn't seen before?
I could not agree with you more. The National Institute of Environmental Health Sciences has a very large database, the name of which has escaped me, but it takes into account environmental factors and human factors and socioeconomic factors across the nation, trying to integrate that. And especially when you get into issues of, you know, what are the chemicals in the water supply and the soil and so on. And I think you're right that we're going to need all the AI we can get to try and collate all of this information to pinpoint factors that are acting in concert to lead to toxicity. And the examples you give of the sudden explosion of exposures in the setting of, you know, inhalation injuries around all of those examples really reminds you how complex this is likely to be. And it may be that there is a 1% contribution from 100 different compounds, which will make it really hard to pick out. And we're going to need a lot of computational support to try and do that.
Yes, please.
So my research is actually on the topic we are discussing right now, is on the integrative approach to investigate inflammation among the African American pregnant women. And I'm trying to integrate the social environmental factors, biological factors, like omic profiles and the inflammation, how they are interrelated. And what we have found out so far is that some inflammation factors are related to the obesity and other factors. And some of them, like CRP, which is a systemic inflammation marker, is highly related to the lipidome metabolomic profile, but is not related to the obesity. It's more related to the depressive symptoms. And the next part of my research, which is I'm funded right now from K-12, from Bishar, is on the PFAS and other environmental exposures, how they're affecting specifically this population, which have a lot more adverse pregnancy outcomes and other things like that.
Dick?
Yeah, I would say to John and to the other comments, it's really about funding. That's what moves science, and the VA is willing to fund. I'm sure the DoD funds this area. My colleagues in Europe have said that their funding is now moving towards climate change effects on disease. And I know that Harvard just started an institute related to this. So I think as we run into more of this and realize that it's impacting disease, the funding will follow, and then research will continue on that. But I think that that's going to take some initiative and some of us initiating that process, if we think it's a very important aspect, and it is. Right?
Yeah, John, correct me if I'm wrong. I think I just read that there are now more people, like the first responders and people that were close and exposed to the 9/11 aerosolization have now died than actually died in the towers that came down.
Yeah, that just came out last week. But yeah, it's a good example, I think, of just kind of what you said. We're looking at potentially new diseases, and funding needs to follow. And what can be frustrating is that sometimes your grants, your ideas are held up against what is known about all the established diseases, and you feel like you're barely scraping the surface of something, and they're like, well, that isn't state of the art, but you're in a brand new field, and you got to start somewhere. So I'd love to see some of that shift in funding take that into account.
So, John, do you know the statistics? Were most of those people that died of the secondary effects, was it cancer or was it like fibrotic disease? Did they have that breakdown?
Boy, that's a question I don't know the answer to.
Any other questions? Okay, this one is for Dr. Holland, which I think is a very good one. What opportunities do you see for partnerships between intramural and extramural investigators, and what things can the NIH do that a university might not be able to do?
Well, there's not much that we can do that you can't do here. This is an extraordinary campus with some outstanding investigators. I think what we can do, though, is that we have the privilege to do some things that we want to be able to share. One of those is that we have this building, the clinical center, and everything that happens there is covered. That is, there's no insurance, there's no citizenship need. It's all about medical need. And the medical need is not just severity of illness. The medical need also means the importance of the study. So we can do some things on patients, either inpatient or outpatient, that would be very hard to be able to do in an extramural environment. We don't need to get permission to do a scan or those kinds of things. So that's something that we can do. And so we can help with deep phenotyping, with complex care. And there are some diseases that we're frankly specialists in that we've seen hundreds or thousands of that most people don't get to see because they're so uncommon. So that's one thing that we can do. In terms of how to work together, there are several ways. Of course, collaboration is open and free in this country, and we're very lucky to have it that way. But it requires funding, and we've already heard about that. One of the funding mechanisms that helps a lot to move back and forth between the extramural and the intramural community, there's something called a U01 grant, and these are opportunities that allow extramural investigators to get funding. remember that we, intramural investigators, we can't get funded on extramural sources. So if there's a U01 grant, it gets written predominantly to come to your laboratories. And those are things that we can work on together and be, co-authors on. There are also collaborative opportunities. We intramural investigators can collaborate on R01 grants, but we don't take any money from it, so it's sort of a win-win. And then there are things that we can do informally at any time. So I think there are a lot of mechanisms, and then we need to look for new mechanisms that allow us to interact more directly with our colleagues here and around the country. I think I just saw the data this morning about the rate of publications that come out of our intramural program that have extramural authors on them, just as a sign of the collaboration. It's about 50% of the papers in our intramural community are really done in collaboration with extramural investigators. So, we're very open and very enthusiastic about collaboration. We look forward to it. But I would encourage you to look to us to help with some of the complex or really difficult medical problems that you might have that we could be of special service for. And I'm happy to help give you advice about who might be the right interlocutors in the intramural program.
Great. I wonder if you could speak a little bit to the training opportunities for postdocs that could come to the NIH?
They're amazing. We've got laboratories, we've got basic laboratories. So one of the drawbacks, and there are several drawbacks at the NIH, one is that we have a fence around it, which really detracts. And I walking around Ann Arbor, I'm struck by just the vitality of the community. And you have vitality for many reasons. Number one, you've just got a great community here. But the other thing you have is a medical school and a graduate school, attached directly to an undergraduate campus. And that vitality is really important. We don't have that, so we need to be able to interact with people on the outside. We can collaborate in granting PhDs, but we can't grant them ourselves. So people could come from institutions. So we have collaborations with Oxford and Cambridge, where we have students that go back and forth. They grant the degree. We provide some of the laboratory opportunities. And we do that with other institutions around the country through what we call the graduate partnership programs. But aside from that, people can come and do periods of training or collaboration in our laboratories, all as part of their training here, as well as come and do rotations if you're a student, either a graduate student, undergraduate student, or a medical student. And then we've got fellowship opportunities in, endocrinology, infectious disease, immunology, and so on. So there are a lot of training opportunities there. It is really I think we are the Rodney Dangerfields to some extent. Everybody's heard about the NIH. Very few people have actually been there. And even fewer know that we have a hospital that we would like to train young people in and to be helpful in terms of taking care of patients in.
Great. So I'd just like to give a shout out. I've had an honor of serving on the scientific board of counselors for the Steve's Institute, the intramural NIAID, and as part of the interrogating and talking to the individuals, that we get to talk to the postdocs. And Steve's right; they have some amazing postdocs. These postdocs have amazing opportunities to have access to resources that we often don't have here, access to clinical patient samples that we sometimes do and sometimes don't, and it is an amazing opportunity there. So thanks, Steve.
Can I ask a question? So you talked about U01 opportunities. So those U01s, are they reviewed by normal study sections or are they reviewed by NIH panels?
There are a couple of different kinds of U01s. There are the ones that are reviewed by the regular study sections, and then there are ones that are reviewed specifically by each institute. So there's another group that are reviewed by the clinical center itself. So there are several different kinds, and I don't remember their numbers, But there are several different ones, and you can go to the website and look at which one is available which year. We don't have them every year, but the typical UO ones that go through the institute or the clinical center are for $500,000 a year for 5 years.
Thank you.
Any other comments? So we saved the easiest question for Nick, is what do you see as the future of inflammation research at the U of M, and where do you feel that our strengths are, and where are we going?
This one I get in trouble, right? So first of all, I wanted to thank all the speakers because they exemplified exactly, I think, where our opportunities are here. To differing degrees, they presented data from patient populations and of modeling. And really, I think those are our opportunities here. We have extremely strong bench research, and I think that's displayed in our NIH funding, which is, I think, predominantly R01 funded. We have a lot of R01s here. We have fewer, as most of the leaders will tell you, we have fewer large grants, right? We have fewer U19s, PPGs, clinical-based interactive grants, and I think that's where our opportunities for growth here. But it takes commitment, right? It takes commitment from the preclinical folks like I am or foundational scientists, I guess, which we have a lot of strength in. We have strength in clinical research, and it's bringing all of us together. And it takes commitment from the institution in protecting time, especially in our colleagues on the clinical side, where the demand is very high for patient visits and really commitment for them to have protected time to do the clinical research. So I would say that our opportunities for growth and opportunities for future in inflammation as well as other areas is in the collaborative between our extreme strengths in preclinical and foundational research and with our folks on the clinical research side. And that commitment, again, takes some funding, and U of M in general has produced funding or given funding to try to get that to work harder or work better. But team science is really within the individuals. It's within our control to interact with our collaborators, to initiate those, and to find the time to actually do that.
Yeah, Vince?
Yeah, Nick, to get to that point, though, of the team science, how do we train the Ph.D. postdocs to be able to communicate with the clinicians? How do we train the pure ND clinicians who have not had a lot of research training to be able to recognize and be able to kind of collaborate back the other way?
Yeah, it's a difficult question, right? Because again, it's individual. Several years ago, there were workshops that were put together to try to talk about team science and how do you build good teams. It's a lot of interpersonal relationships, and that can only happen as the mentor to really help initiate that. There are programs here, translational programs here at MISHR. There are some transitional T32s, and it's really some commitment for the folks that are running the T32s to help bring that together, I think, because that's really an avenue that we've used. A very strong number of T32s here, and those folks can help. The K-12 at MISHR is very helpful in that. But I think it becomes effort on our part as PIs and mentors to help initiate that. As an MD researcher yourself, you guiding those PhD and postdocs, towards interacting with those folks is going to be really important. So.
Turn it to Steve.
Yeah.
How can they help us?
Money.
But I'm asking you to ask Steve for how he can help.
Steve, how can you help?
No. Give him an idea.
Yeah, truly, we're open to ideas to fund for sure. And one of the things I'd just like to ask Nick, I know you are one of our stalwarts studying virology and virus interaction and pathology. So is that still an area that you think has fertile ground for study?
Yeah, we just started, or the University of Michigan just started a cross-campus center on, infectious disease threats. There's many individuals here that are studying virus infections in many contexts. I think that there are still opportunities in funding in looking at viruses, but those stretch across more, we can talk to the other panelists who do tumor research. There's many opportunities in tumor research and to build these cross translational communities. But that takes time, it takes commitment, and it takes more than just commitment from one side or the other. I think it's something that we all have to initiate and try hard to do. And I think it became harder once we went to COVID, right? That disrupted anything that was happening. And as we were talking earlier, getting those folks back and getting them engaged in person opportunities like this, I think are very important.
Yeah, to put an exclamation mark, I don't know if you heard the interview with the individuals who won the Nobel Prize for Medicine and Physiology, and they asked the woman who's Hungarian, and it's Kariko, said, you know, you interacted with Drew, where did you first meet him? And she said, over the Xerox machine. I was Xeroxing some things, he was a new faculty member, and we started a discussion about what we were doing, and she said the next thing I knew, we were collaborating together. And we need to all come back, because that's where innovation is missed, over the water cooler and the coffee pot, and just meeting each other in the hall again. And I think that's one area that really underscores why we need to get back and interact with each other. So I'd like to ask Weiping one thing about the research focus. What's, you know, mRNA therapy has been really just jump-started and was game-changing and saved millions of lives. But people keep thinking about mRNA therapy as vaccines for tumors. How viable do you think that's going to be?
Yeah, that's, so first of all, I have to say I do not work on mRNAs, so therefore anything I say, I don't know if I have any direct experience on that. But saying so, actually, as far as I know, a few companies, including Moderna themselves, they are working on mRNA vaccines. So far, they want to combine. So the thing is, what we basically think, any single or monotherapy will not work in the cancer field. So that's the common sense in many ways. So in this case, it's possible a mRNA vaccine may not work as a monotherapy, may be combined with other depths of therapy, including immunotherapy. How do we do that? And I guess the foundation is really the basic research. We need to understand much better how the immune-associated microRNAs are regulated, degraded, controlled, and in terms of the stability and some enzymatic activity. All those things could be the way to go. And I don't think we can foresee those things. You know, we will see the results immediately. Even in immunotherapy, we talk about this at these days. Since it's a huge success. But even in PD-1, PD-L1, we were involved very early on those studies. It takes more than almost 20, 25 years. So that's why we, on the one hand, we have to invest in this area, but on the other hand, we have to be patient.
Yeah, thank you for that. David.
While we're circling back, we think the questions. I wanted to know, what do you think about the future for bi or tri-specific antibodies when you're talking about immune therapy?
Yeah, so are you talking about, you are talking about the antibodies, right? So the bispecific, tri-specific, is that what are you talking about?
The ones that bring T cells to the tumor, right? And then recognize the tumor with the other arm. Yeah. So I'm wondering what you think the future is with those.
Yeah, so the good thing is once the industry is involved, it is always a good thing because you need to have a lot of investment. So in this case, in this area, there are at least, I don't know how many companies are involved. Basically, people have, even the PD-L1 antibody, CD4 antibody, people start to design bispecific, tri-specific, even more specifics, you know. So this all we need to antibody engineering. In this case, so far as I know, I do consultation as well for some companies. We don't see huge breakthrough in that space. But I feel that's a direction to go. So on the one hand, you want to make sure the T cells can get into the environment, But on the other hand, you want to make sure the T cells are really engaged, can survive, can fight in the tumor microenvironment. So therefore, people are making efforts on this through different angles. I guess we just have to be patient in that space. I feel we will have more product in that space.
But if I may add a comment, in addition to what you said, you also need to deal with the immune suppression in the microenvironment. So we need to take, as you said earlier, it's going to be a multi-pronged attack. I don't think that one particular technology on its own is going to do it.
Yes, you're absolutely right. Actually, the immunotherapy basically deal with the immune suppressive mechanisms. That's the basic focus my research group has been working on. So in fact, in this area, and as I mentioned early on, so people are still looking for different checkpoints, different mechanisms. My gut feeling is basic science will bring the products. So as far as we don't understand what's going on, or we don't have sufficient knowledge in that space, we cannot move too far.
David?
Thanks. It sounds though like if you were going to do bi-specific antibodies, you may need tri or quadra-specific, right? So something that turns off TGF-beta or whatever also.
Yeah, good. Any other comments or questions? Yeah, Pierre.
Why don't you go first and I'll grab a mic.
Oh, sure. Well, since we've got tumor immunotherapist and infectious disease specialist on the panel, I'll ask a question about, there's a lot of chronic infections out there that act kind of like tumors. They suppress the immune system, they evade the immune system. And in tumor immunology, we realized that pouring gas on the fire didn't work, but taking the brakes off did. So will we see things like checkpoint inhibitors used in chronic infections in the future?
Great question.
So, when we talk about cancer, we have to be careful to talk about specific cancers, right? So melanoma is not the same as glioblastoma, not the same as colon cancer. And the same thing's true in infection. And so this question, and boy, it's an incredibly critical one about how do we manage chronic infections. So early on, many of us were bound up in the idea exactly as you articulated it. If we just take off the brake, the car will go where we want it to go. Ironically, in particular, in probably the world's greatest, important chronic infection, tuberculosis, many of us thought, okay, that's going to be easy. Wrong. And when you block PD-L1 in the case of tuberculosis, it makes it worse. And if you block it in the mouse model, it makes it worse. And you block it in the primate model, it makes it worse. And sure enough, because somebody was bound to do this by accident, when you block it in the humans, it makes it worse. So, and I think and then it gets even more complex, and I think really cuts to the theme of today's session, and goes back to the discussion about lupus. There are times when it's like the three little bears. You just got to have the right amount of inflammatory response. And when you block PD-L1 in the setting of tuberculosis, you make things worse. And that's because I think you get too much of that interferon response, and it's lethal. It can be lethal. So we're still trying to figure out which infection is the one that is most responsive to blocking those checkpoints. I don't doubt there will be other checkpoints, and the general point you made about how there are clearly effects of each chronic infection in terms of subverting what should otherwise be an effective immune response. That's really true. Unfortunately, I think each pathogen probably has a slightly distinct way of doing it. So, the pox viruses actually encode proteins to block interferon signaling. CMV does something somewhat similar. Mycobacteria block intracellular interferon signaling through pathways that we're still trying to sort out. And I think we're going to have to identify each one of those mechanisms to figure out which of those checkpoint inhibitors will actually be felicitous as opposed to more deadly. And so that friend-foe dichotomy really turns out to apply, I think, very clearly in the setting of infections and chronic infections and then how to deal with them.
Pierre?
Steve, I'm here now with the microphone. So first I want to thank you, Steve and Marshall, for putting together this lecture series and for picking inflammation and immune response as the first topic. That's really phenomenal. I really enjoyed the half day. I would like to invite you to make a comment. I was in the poster area a bit earlier. It's amazing. This is just a subset of research relevant to these areas that's going on at University of Michigan. So from your perspective, what are some ways that you're thinking about that can sort of magnify the might of our inflammation and immune response research and bring us together? What's coming down the pike to invigorate that area that's already strong and important on campus?
Right, that's a good question, which gets to the point that Kelly Kennelly asked me to remind everyone that there are sign-up sheets in the back for those that would like to be on some of our subcommittees to answer those questions that Pierre just proposed. We do have a lot of strengths here in immunology and inflammation, and where should we place our bet that where the field is going and where we would like to skate to where the puck is going to be. Great question, Pierre. We're open to a lot of discussion with you all, which is one of the reasons why I wanted to use this session as a kickoff into one of the final areas that we will be making investments with regards to those 5 areas where we want to create that a community of scholars going forward. So I don't want to have Steve Kunkel put the stamp on where we should place those bets and who we should invest in. So I'd like to engage our research community. But thanks for that question. Any other questions or comments? So if not, I'd like to thank all of you for taking time out of your schedule this afternoon to join us. I'd also give another shout out to Kelly Kennelly, Ann Curtis, Dave Madrigal, Aaron LaRoe, and other people in Rose Glen Shopping Communications. I'd like to thank our speakers, Michelle, Beth, and Sonia. I'd like to thank our panelists, and I would really specifically like to thank Dr. Steve Holland, who really put an exclamation point on this afternoon you made today, Steve. Thank you.